PTC, phenylthiocarbamide. It's a substance you can either taste or not. For people who taste it, it's an unpleasant kind of bitterness. And it's something that generations of biology students have sampled in genetics lessons because of the way that it's inherited in classic Mendelian fashion, with tasting being the dominant trait and non-tasting the recessive. If you taste the PTC on a little strip of paper, you're either big T homozygous or heterozygous. If you can't taste it, your genotype is little t homozygous, little t, little t. In this lab, we're going after the underlying biology at a molecular level. We already know this as a single gene polymorphism. It's inherited as a classic Mendelian trait with two alleles, big T and little t. But now, looking at the sequences of A, C, T, and G in these two alleles, shown here as non-taster in light over taster in dark, there are three differences at single base pairs. We use the term SNP, which stands for single nucleotide polymorphism, and we usually say SNP. There are three SNPs, or spots in the gene where the alleles are showing different letters. At this position, tasters have C, non-tasters have G. Over here, tasters have C, non-tasters T. And in this third position, tasters have G while non-tasters have A. For all the rest of the letters, the two alleles are exactly the same. These sequences of the same gene show you where the SNPs are. You might also note that this read of our gene starts with ATG. This would be the AUG start codon for methionine. And it also ends with TGA, which makes the stop codon of UGA. This side-by-side comparison of the two genes has been edited to contain only the translated part of the gene. No introns either. They have been edited out. The sequences you're looking at are pure translatable codons. If you wanted to, you could use your knowledge of the genetic code to get the entire amino acid sequence of the gene product, and in doing so, figure out how these three SNPs actually affect the protein's primary structure. We've saved you the trouble here. There are three amino acid substitutions. The protein products for taster and non-taster are the same, except for three amino acids at three different points in the overall sequence, which, if you're counting, has 333 amino acids in total. In the taster, those three amino acids are proline, alanine, and valine, and we call this the PAV allele, whereas the non-taster has different amino acids, alanine, valine, and isoleucine, and this is the AVI allele. The protein we're making here has the catchy name of TAS2R38. This is a protein that sits on top of the membrane of taste receptor cells in your tongue and transduces a signal that gets sent to your brain, allowing you to taste bitter. And there are actually 43 different versions of this particular tasting protein scattered throughout your mouth and throat, mostly on your tongue. Each a protein coded by a different gene, and each one sensitive to a different bitter compound. The one that PTC either triggers or not is number 38 in this set of 43. TAS2R38. The alleles we called taster and non-taster, big T and little t, before, are now PAV and AVI, two versions of TAS-R38, having different sets of SNPs and making a protein that's either alanine, valine, and isoleucine, or AVI, the non-tasting allele, or proline, alanine, and valine, or PAV, which does interact with PTC and causes you to taste the bitterness. Now let's go through some questions to make sure you really understand what's going on. You can back up and replay the video before answering. You only get one shot at the right answer, so make sure you're not just guessing. Okay, now comes the practical part. The nucleotide that's there at any one of these three SNPs should be enough to tell you if you have one form of the gene or the other. What we're going to do in this lab is to collect our own DNA and focus precisely on the second of these SNPs. We're going to find out if it's either C for the taster allele or T for the non-taster. Think about this for a minute. There are 6.5 billion base pairs in our diploid genomes, and we're going to zoom in on a single base pair in this analysis. Obviously, we're going to need some pretty sophisticated methods and equipment, And the procedure you follow in order to complete this successfully is multi-step. Now you must pay close attention to the written instructions and follow them to the letter. As in almost any multi-step procedure, if you mess up anywhere in the sequence, you won't have any results to look at later. What I'm going to do with this video is to walk you through the whole lab to give you the big picture. Understanding what you're doing and why makes a huge difference for the success of this lab, both in terms of the results we get and as a learning activity. Now, the written part of this lab explains things really well, and you should read through the procedure carefully and twice at least before you come to class. All this preparation will actually save you time later when you find that you can carry out the steps with confidence rather than having to reread at the last minute because you're not sure if you're doing something wrong. So in this big picture, there are 4 major steps involved. In step 1, we get DNA from our cells. In step 2, we amplify just the part of your DNA with the TAAS2R38 gene, that is the region that contains the SNP in question. This is what we do in the first week. In the second week, we start with step 3, which is to use a restriction enzyme to break the DNA or not, depending on which version of the SNP is there. In step 4, we use electrophoresis to see if the DNA has been cut in two or not. So the first general step is collection and isolation of your DNA. You start by collecting cheek cells, and you'll do this by rinsing your mouth with salty water and spitting into a tube. After spinning down the cells in a centrifuge, you'll pour off the supernatant and transfer the cells into a little tiny tube that contains calyx resin beads, and then you'll mix this in a vortexer, which is a vibrating rubber plate. Then you'll spin down the contents of your little tube in a microfuge, and by this point your cells will have first been thoroughly mixed with the resin beads and then repelleted with a microfuge step. Now, you'll cook your cells in a heated block and then mix it up again in your vortex so that everything in the tube, cell stuff, and resin beads is thoroughly suspended. This is where you'll spin things down to the microfuge again. Now, between cooking the proteins into solids and the resin beads attaching to the divalent cations, all of the undesirable parts of the mixture will have been spun down into the pellet, leaving the DNA in the supernatant solution. Now, this step, it's crucial for you not to get any of the pellet. The cations that are captured on the resin beads will interfere with the next step. The second general step is to use PCR, polymerase chain reaction, to make lots and lots of copies of just the region we're interested in. We use custom-made single-stranded DNA primers to serve as the bookends to define the specific part of the genome we want to copy. Basically, we're telling the PCR enzymes to copy here while ignoring the rest of the 6.5 billion base pairs. Now, PCR is possible only because we have special enzymes from the heat-loving microbes like Thermus aquaticus. This is the source, the Taq polymerase, used for PCR in laboratories across the world. The PCR thermocycler is basically a robot that changes the temperature between warm, hotter, and hottest in the precise way such that the primers of the DNA polymerase can do their magic. All you do is to put your DNA, the enzymes, the primers, and also lots and lots of mononucleotides to serve as building blocks for the DNA synthesis, and then just let the robot do its thing. By the end of the first lab, you'll get this started before you go, and then someone else will collect the samples and store them in the freezer until you come back the next week. Steps 3 and 4 come in the second week of laboratory. You'll get your PCR mixture back, and then you'll take 2 samples of 20 microliters. This is a really, really small volume. And to one, you'll apply the restriction enzyme FNU4HI, and the other one won't get the enzyme treatment. Enzyme FNU4HI comes from the bacterial species Fusobacterium nucleatum, and it makes a double-stranded break in the DNA anytime that it sees the sequence GCNGC, where N can be any of the 4 nucleotides. Now, if you look at the second SNP in the handout, you'll see that the taster sequence has GCTGC, while the non-taster has GTTGC. In other words, FNU enzyme is going to break the taster's DNA at that point, but it won't break the DNA of the non-taster. Consequently, the digested sample of the taster's gene will have 2 shorter fragments of DNA, where the non-taster as well as the undigested sample that didn't get the enzyme will show just a single longer fragment. By the way, the non-taster should show no differences between the digested and undigested samples because it lacks the restriction site. The last step, step 4, is where you load the samples into an electrophoresis gel and then put an electric current through the gel such that the DNA migrates through the gel to different distances based on the size of the fragment. The distance traveled is inversely proportional to the size of the fragment. Smaller DNA fragments travel farther. Loading the gels with the samples is kind of dicey, and it takes a very steady hand. You want the sample to go into the well, minimizing spillage outside of the well. And this is made just a little bit easier by the loading dye that you will mix with your sample before loading it into the tiny slit in the gel. Now the loading dye does two things. It's blue, so you can actually see your sample as it's going into the well. And also it contains a bit of glycerin, which makes the sample just a little bit denser than the buffer that's already there in the well. And this helps just a little bit to get the sample to sink gently to the bottom of the well. Oh yeah, I forgot to tell you that the gel is actually below the surface of the electrophoresis buffer that's there in the gel rig. Now when you load the well, the one mistake you want to avoid is shooting the sample out of the pipetter with any force, because that will just blast out what's ever there in the well. Big mess. The other thing we do is to put a DNA ladder into one of the wells of each gel. This contains a mixture of DNA fragments of known length, producing a known pattern in the run, and so it makes basically a ruler on the gel by which you can estimate the sizes of whatever fragments of DNA are in your gels. Note that with the electrophoresis results, you should be able to distinguish between heterozygotes and both homozygous genotypes. Homozygous for the non-taster allele, AVI, should show only a single band that's relatively long and nearer to the start, and about the same length as the undigested DNA. Homozygous for the taster allele, PAV, should show two bands that are both shorter than the undigested DNA. Now, heterozygotes will have one copy of both alleles. In other words, they'll have that long unbroken fragment as well as both fragments of the digested DNA from the PAV allele. You'll see 3 bands in total. You'll document your results with a photo of your gel with a complete interpretation. How the digested sample compares with the undigested sample, how many bands were there in the digested sample, what were the estimated lengths of the fragments based on the latter, and do the lengths in the digested sample add up to the lengths of the undigested DNA? There's also going to be class-wide data that we collect in comparing these results with a direct phenotype assessment that we do of ability or inability to taste PTC from the paper. And this concludes the video of your pre-lab for the genetics of PTC tasting.